Good evening, everyone. I hope that this shot of daisies in a forest that I took a couple of years ago, it's not a great shot, but I hope that it might express or convey to a large extent how the current COVID pandemic has made us realize the value of the great outdoors, bringing home the necessity of working together for a better future, for preserving Earth's natural resources and addressing the various global challenges that are facing us: urbanization, deforestation, finding ways to feed an increasing population, fuel and food security, and above all, climate change. As many of you might be aware, much of climate change is driven by the rising levels of atmospheric carbon dioxide. And these levels, currently at roughly 400 parts per million volume, are higher than what Earth has seen in the last 800,000 years. It's true that the current COVID lockdown has brought much of these emissions to a halt for the time being. But who is to say that the world will not, or we will not, be speeding right back into our jet planes the moment we get a chance? I do hope that some of us, at least, are able to reflect upon the carbon footprint that we leave and how it takes tens of thousands of years for all that carbon, all that emitted carbon, to make its way back into the Earth's crust. Leaving aside the causes of this increase in atmospheric levels of carbon, what we need to do urgently is to find ways of bringing it down. And this is where plants come in. I'm Gita Yadav, and I'm a plant scientist. I work at the Department of Plant Sciences, Cambridge, and I also hold a joint appointment in India at the National Institute of Plant Genome Research, New Delhi. Today, I shall be talking about green carbon for food security. Even though many of us are aware that plants are the natural air filters of the earth, we still fail to appreciate the true value of the natural capital of plants and of photosynthesis, the process by which plants are able to A, draw down atmospheric carbon, B, fix inorganic carbon into organic carbon, the carbon that all life forms are made up of, otherwise known as food, and C, release pure oxygen back into the air. As researchers, the questions that we are asking are: which plants are most efficient at photosynthesis, and where do they live? And more importantly, can we understand them better to improve other less efficient plants? Surprisingly, the answer to the first question is in a tiny unicellular green alga that lives all around us, mostly in freshwater habitats, but also soil environments, the ocean, snow, you name it. Ocean phytoplankton make up less than 1% of the total plant biomass on Earth, but they are responsible for nearly half the net photosynthesis going on this planet. Research has shown that green algae are able to fix carbon up to 40 times better than higher plants. To understand how they're able to do this, you've got to first understand the issue of efficiency in photosynthesis altogether. The key is the manner in which these little green plants have managed to rectify a flaw that is at the heart of photosynthesis. And the flaw is this: Rubisco, the global carbon fixing enzyme, the guy that stands at the interface of living and non living, performs less well in most plants due to its inability to distinguish atmospheric carbon dioxide from atmospheric oxygen. And plants usually respond to this situation of wrong number dialing by making huge amounts of Rubisco, so much so that it is the most abundant protein on Earth, and yet it's not enough. How would it be when oxygen levels are much higher than carbon dioxide in the air? So, back to the question: how do algae fix this flaw? With this little background, I'd like to introduce you to the model unicellular green alga that we are working with in the lab that fixes carbon very well, and it's called Chlamydomonas reinhardtii. This is the tiny little plant form that we are working with to understand the business of super efficient removal or fixation of carbon from the atmosphere. So the ingenious trick that this little beast, Chlamydomonas reinhardtii, or Chlammy as we like to call it in the lab, has evolved is to physically separate Rubisco from oxygen, resulting in a tremendous increase in photosynthetic efficiency. Chlammy does this by packaging Rubisco into a tiny little microcompartment called the pyrenoid that you see here in the figure. The pyrenoid is chock full of Rubisco and is designed in such a way that while carbon can enter it and get fixed, Oxygen cannot, and thus, by proxy, Rubisco sees only the correct substrate rather than oxygen. Such mechanisms that physically separate Rubisco from oxygen by concentrating carbon dioxide around Rubisco are called CCM, an acronym for carbon concentrating mechanisms or carbon dioxide concentrating mechanisms. And these have evolved not just in clammy, the model organism that we are studying in the lab, but also some blue-green algae, as well as some higher plants like the C4 and CAM plants. What we are trying to do is to identify key components of the clammy biophysical CCM in the hope of using it to improve higher plants and crops. And this brings me to the work that we're doing with my collaborator and colleague, Professor Howard Griffiths at the Department of Plant Sciences, Cambridge. Our research is to interrogate very large genomic data sets and find molecular fingerprints or patterns in the DNA or RNA that may help us in identifying finer details of this process. In our case, the algal carbon sequestration. So what we've done basically is to collate over 50 important genes that we felt were important for the CCM from several decades of published literature of various kinds. And then we took these core set of important genes and divided them into time segments as shown over here, based on when these genes are expressed during the day-night cycle of clammy growth. So in this picture, you can see a heat map with the green colors showing the genes that light up or get activated at various time points in the clammy day-night cycle. And then for each one of these time segments that you see in the heat map, such as you might have cell division followed by resting stage, the pre-dawn that happens just before the onset of light, all of these in the dark, and then active CCM in the first 8 hours of light and so on. What we did was to extract the genes that are switched on in that time segment. In other words, all the bright green colors that you see over here in each vertical box, we pull them out and then we build gene correlation networks to understand how the genes in that time segment are associated with the remaining 17,000 odd genes in Chlamydomonas. Most of this work has been done by my PhD student C2. In this slide, you can visualize the clammy CCM gene correlation networks that C2 has built. And what we observed, and you can see in this figure, is that there are critical regulatory subnetworks that are active at different time points. Most importantly, we find that there is a unique set of regulatory genes that are only active during the hours just before dawn. Take a look at the second network. It's called the pre-dawn network, and you will notice a bright orange shaded node, quite a few bright orange shaded nodes. Each dot is a gene essentially, and these guys are together switched on only during this time segment specifically. Since this activity takes place a few hours before the onset of light, and that's when active CCM kicks in, we are now in a position to explore this little set in the pre-dawn network in more detail to identify the missing links in the regulation of clammy CCM. In the long term, we hope that some of our findings from molecular research may be translated to the field or to higher plants, even crops. Much of this work is funded by the TIGR2ESS Program. It's a Global Challenges Research Fund Grow Award for transforming India's green revolution by research and empowerment for sustainable food supplies. It's quite encouraging for us that some of our collaborators at Princeton and York University have been able to successfully get some elements of the biophysical CCM into higher plants, like tobacco. For me personally, the TIGR2ESS program has been an amazing roller coaster ride, encompassing much more than cutting-edge research in green carbon. TIGR2ESS has been a fabulous learning experience, bringing home the amazing benefits of working at the intersection of multiple fields and technologies. Between 6 multidisciplinary flagship projects that we have in this program, over 30 different organizations across India and UK have come together as TIGR2ESS partners, and we've joined hands across disciplinary silos of natural sciences and social sciences. We use high-resolution land records from rural India. We use genomes, as I do in my work. We use fossil records of pollen in ancient civilizations. We use supply chain modeling, agricultural economics. We talk to farmers. We work with women's collectives. We use robots, geography, mathematical simulations, and all kinds of data analytics to address the global challenge of feeding an ever-increasing population. Most importantly, within TIGR2ESS, we teach and train rural and urban youth, and we contribute to capacity building as well as equality and diversity. On that positive and forward-looking note, I'd like to end my talk and thank everyone who continues to enrich my experience as a plant scientist. I thank my students, colleagues, friends, mentors, college members, St. Edmund's College, Cambridge, and collaborators in both India and the UK. I especially thank the Cambridge University Botanic Garden and the Camp Plants Hub for suggesting that I do a research short for the Science Festival. I thank our funding agencies in both nations, the program managers and TIGR2ESS, very much so. And thank you all for listening. Signing off now, Gita Yadav for the Cambridge Science Festival. Thank you.